Dhugal Lindsay from the Japan Agency for Marine Earth Science and Technology here, talking about requirements for imaging surveys of pelagic communities. I'm currently taking part in the first environmental impact assessment in the Clarion-Clipperton zone of the midwater community using ROVs. So I have some insights from there. This is a frame grab from the 4K video camera from the ROV we've been using, and I'm sure you can all see the animals in this picture. No? Let's brighten it a little bit, and I'm sure now you can see the animals. No? Here are the animals. They're kind of hard to see, so we'll zoom in a bit, and this is the original resolution. Now we have 3 animals here, and I'm sure you can tell what species they are. Nope. Well, there's a trick. This is video, so we have 30 frames per second, and we have 3 frame grabs here. The original video is kind of hard to see. We brighten it up in post-processing, and we see this long snake-like transparent animal in the 3 images. It has a repeating white, clear, white, clear, white, clear structure. These are the chlamydia, the repeating units in the tail, the cyphosome of the siphonophore. And you can see around the head of the siphonophore here where the swimming bells are. In this image you can't really see anything much, and then suddenly these bright spots appear 33 milliseconds later and then disappear again, and there's one in a different place. So what this means is there's a reflective structure which is moving rapidly on this end of the animal. The cyphosome meets the swimming bells here, and we can see the reflections on both sides of the start of the cyphosome, so we can tell there are 2 swimming bells, and there's no bright white structure here, which would be the float, so it's not a physonect, it's a chalicophorium with 2 nectophores. You can also see, if you look very closely, these curved reflective structures, which are the edges of the swimming bells. So I'm able to tell you that it's a prayed siphonophore, and the clearest looking cyphosome means it's either Liliopsis or it's Desmophes. Because the reflections are appearing here on the swimming bells, I can say it's not Liliopsis because the light hitting the swimming bells would reflect off in this direction, not back into the camera. Desmophes, the edges of the swimming bells, would reflect the light back into the camera, so I can say this is the genus Desmophes. So that's how we need to identify animals. It's not really important how we identify them. The take-home message is we need really good video, and we need side lighting to get the highest contrast we can for mostly transparent animals on a black background. Try to lighten them up and keep a lot of contrast with the background. We need very high resolution so we can see all the fine structural details of the animals, and we need fast frame rates because we use the behavior to help with identifications. In the previous example, it was seeing how the reflectivity changes, so knowing there's a structure there even though we can't really see it. Sometimes it's the actual behavior of the way the animal escapes. They look the same when they're out of focus and far away, but they escape in different ways so we can tell what animal they are. So we need 4K video or better. It's best if there's 60 frames per second rather than 30, but more important than that actual frame rate is that it's recorded in progressive mode rather than interlaced mode. In interlaced mode, because you're recording one field and then another field and then a field again, you get this disparity as an animal moves across the screen, where the edges of the animal take on this spiny comb-shaped appearance and you can't see the fine structure anymore. The other thing is, even if the camera is a 4K camera, unless you do your recording in something like an Apple ProRes 422 codec, you have compression artifacts. So you can have a nice image coming through the camera, but if you record with lots of compression, the image is no longer good for analysis. This is really bad in the black end of the spectrum, where most cameras say, oh, it's a black background, we want to make it as black as possible. So any small changes in the black is all made into a single black color, which makes it really hard to see a transparent animal on a black background if you use one of those high compression codecs. So if you do all of this, we're able to identify the animals to species or genus. But if we want to quantify things, it's another problem altogether. With the benthos, you often shine lasers on the seafloor or on animals. They're like 5 centimeters apart or something, and you can tell how big the animals are. Unfortunately, you have a really hard time hitting the animals in the midwater. Plus, the laser light goes straight through the animals without being reflected back into the camera because the animals are transparent. So along with the side lighting, you need stereo cameras. And if they're properly calibrated, you can then say this pixel in left camera is equal to this pixel in the right camera, and therefore the size of the animal is this big, and the distance between animals is this much, therefore there's this many per cubic meter. This is okay for video. Large animals can be seen in the video, this side of the size spectra. If you want to image smaller animals, there's lots of very small, transparent, soft animals in the midwater. You need a different set of cameras. 2 of these cameras is, one is the shadowgraph camera system, which uses transmitted light, collimates the light so all the light beams are parallel to each other, and then records it on a camera. Why this is good is because if you have something close to the camera, it looks really big, and if it's far from the camera, it looks small. If you're taking a photograph of the shadow of the animal rather than the animal itself, and you're doing it where there are parallel light beams, the size of the animal doesn't change depending on its distance from the camera, so we can get correct size information for animals. The UVP does this in a slightly different way. We know the distance from the camera to the very thin sheet of light, so we know the size of the particles in this case as well. We've done something like using a CTD rosette system. We collect water samples where you can get environmental DNA samples to get DNA sequences from the animals there. We attach different imaging systems to this, like the visual plankton recorder over here, which takes dark field color images like these ones in the bottom left. We also attached a UVP. The red light here allows you to not worry about the background lighting, which is mostly blue in the surface waters because the camera only picks up the red, and you can get these nice, almost shadowgraph-like images as well. This allows you to quantify the particles, how many of them are there, how abundant, at which depth, as well as getting information on other animals that occur there. You can also scan plankton that's collected in a net, and this also allows you to get size information for the plankton, and the images are more easily stored in a database than a jar of wet tissue. For all imaging systems, if they're real time, we need the data to be synchronized with Greenwich Mean Time. So we know that when an imager is imaging a certain packet of water, all the other imaging systems are also imaging that same packet of water. And any environmental data parameters like salinity or temperature, and any platform or camera metadata such as the depth, the altitude, the zoom ratio of the camera, etc., needs to be logged at each instrument's native rate, which means if your instrument's recording at 4 recordings per second, you save it at 4 recordings per second. Once per second, you save it at once per second, and then you munge the data together afterwards. What you don't want to do is collect everything at the same rate as your slowest instrument. If you get everything at 1 frame per second and you're rapidly zooming in and out, you lose information on the size of the animals, for example. So always log things at the instrument's native rate and pin those times to GMT. Make sure they're synchronized. For midwater surveys, videos need to be analyzed. So we have made a video annotation system with a video player that works on Mac, Windows, or Linux computers. You play the video back on the frame you want to annotate, you capture, and it gets sent to a web server. You then annotate the still image that's been captured from the video by clicking on the organism and then typing in the little window the taxonomic name or selecting it from the list. We use a worms-based naming system. You can then tag the annotation. Best shot, is it where the animal entered the frame or exited the frame, is it in focus, whatever tag you like, and then you can draw a polygon or a bounding box around the organism. This is to either inform human annotators where the animal is inside the image, or also to teach a machine what part of the image is the animal for training machine learning pipelines. And this is stored in an SQL database and can be exported in the COCO format so you can directly train machine learning algorithms.